Back into session here. Dr. Wolin is back. He's going to speak on some clinical trials or what's going on in the treatment of NETs. So let's have a seat. We'll get this going so we don't have to keep you too late. I know that it's been an interesting session, but I know you probably don't want to stay till 6 or 7 or 8 tonight. So.
Okay. Thank you. It's nice to be back. Does the mic come off? Hello, is this better? Okay, I'll try it like this. Okay, so now I'm going to be talking to you about what's new for treating neuroendocrine tumors in 2019. Before we were talking about some background information, and now we're going to talk about clinical trials that have recently been done and have totally changed the landscape of treatment, and we'll be talking about upcoming clinical trials which are very exciting. Okay, first of all, as you can see on the slide here, this is a slide showing you a timeline of what we call randomized phase 3 clinical trials, which absolutely proved beyond any question that these therapies were successful and in every single case led to the FDA approval of a new treatment for neuroendocrine tumors. When I started treating neuroendocrine tumors, there was not one drug approved for carcinoids in the United States. Nothing. Sandostatin's never been approved to this day as a treatment of the disease. It's only approved for treating diarrhea and flushing. So we had no disease treatment options at all approved in the United States. And as you could see, beginning in 2011, in rapid succession, we had all of these things that we're going to talk about, all of them, which have led to effective treatments for metastatic neuroendocrine tumors. And I think it's extraordinary how fast this has happened. The number of every one of these is based on results of a clinical trial, and we'll talk about all these clinical trials. And there's a whole new generation of clinical trials going on now. You just heard some a few moments ago that Dr. Del Rivero was talking about, and clinical trials everywhere are going to totally inform the treatment of neuroendocrine tumors for years to come, the trials that are already starting now. And in many cases, the only way to even get treated with anything that has any hope of working is with a clinical trial. So I want to just share some of this with you. The first molecular target I'd like to talk about is the somatostatin receptor and clinical trials that involve that. We've heard a lot about the somatostatin receptor already. Somatostatin is a natural hormone in the body, which is the body's turn-off hormone. It turns off endocrine production of hormones. It turns off the production of neuroendocrine cells, and in high doses can be used as a therapeutic drug. Natural somatostatin only lasts in the body for 2 or 3 minutes, so it isn't a very effective drug. So what is called somatostatin analogs have been made that can last for 8 hours, they can last for a month. These are called somatostatin analogs. The ones that we're most familiar with here are Octreotide and Lanreotide. The action of somatostatin is to bind to the somatostatin receptor on the cell membrane. It's a particular feature on the membrane of neuroendocrine cells that allows somatostatin to stick to the cell. The moment it sticks to the cell, it gets internalized into the cell, because of the kind of receptor that it sticks to, always internalizes. So whenever it binds to a cell, it goes into the cell and it's stuck. And that's where it exerts its action. This is important when we talk about using somatostatin analogs in PRRT or for imaging, because it always is highly localized to the cell. Okay? The drugs are extremely well tolerated. This is from a slide of Dr. Pavel, looking at toxicities of neuroendocrine somatostatin analogs, I should say. And you can see that the most common toxicities are actually gastrointestinal. And almost always these toxicities are the result of a deficiency of pancreatic digestive enzymes. Because what somatostatin analogs do is, in addition to being an antagonistic thing for endocrine hormones and for neuroendocrine cell growth, they also stop the production of digestive enzymes from the pancreas. These digestive enzymes need to be squirted into the intestine in order to digest food. And if you don't have enough digestive enzymes, the undigested food goes all the way through the small intestine into the large intestine. The bacteria party on it and ferment it and make gas, because that's what bacteria do when they eat things. And next thing you know, you're turning into a gas factory and have bloating and pass a lot of gas and are miserable, and also are constantly running to the toilet thinking you have worsening diarrhea because you don't know if you're running to the toilet for a little bit of stool and a lot of gas or vice versa. So that's a big problem. And it's a very easily fixed problem because pancreatic enzymes are available in a pill, the most common brands being Creon and Zenpep. It's called pancreolipase is the generic. And these kinds of pills can be taken before you eat and will provide the natural digestive enzymes that you're missing. So it's more of a nutritional supplement than a conventional medicine, but it really does work. Okay, the other side effects are less common. We mentioned the gallstones that can happen earlier on. You can have a little bit of a pain in the butt where the injection goes in, as with any injection, but it's usually not a major problem. Blood sugar can go up or go down a little bit, but most people tolerate it extraordinarily well, considering how many hormones are being suppressed by the somatostatin analog. And there are people who have been on them for 10, 20, 30 years or more with control of their cancer or tolerating it. There was a study done trying to look at Octreotide LAR as an anti-cancer treatment. It was known as the PROMID study, done in Germany. It was prematurely closed, and it's impossible to interpret it properly. And on that basis, it was never able to be relied upon for approval as an anti-cancer drug, even though it probably does have anti-cancer effects similar to Lanreotide, and it's used pretty widely as an anti-cancer drug, but it hasn't been able to be proven by a randomized trial. That randomized trial that was done in Europe, the people that were treated with Octreotide LAR injections had an average time of cancer control of 14 months, and the people that got no Octreotide LAR had cancer stable for 7 months. So it looked like it doubled the period of control. However, as you'll see with the studies with Lanreotide that were much more comprehensive trials, the period of cancer control was much, much, much longer. So in any case, the big trial that has really put somatostatin analogs on the map as a treatment is the so-called Clarinet trial. I have the letters in yellow that spell clarinet. You really have to use your imagination to make clarinet out of that crazy title. But in any case, this is the Clarinet trial, a randomized trial of Lanreotide versus placebo, done before there was a single drug that was approved. As you saw in that first slide of clinical trials, at the time this was done, placebo was a perfectly ethical thing to do because there was nothing else to do that was available as standard of care. So half the people got nothing and half the people got Lanreotide. But the way the study was written is that if anybody progressed who was on the nothing arm, they were able to cross over and get real Lanreotide. So it was a good trial and was gave remarkable results. So these are the results that were published in the New England Journal of Medicine when we first published this. The people who got Lanreotide, the time until the cancer started growing again was not even reached. Most people still had control at the time of publication. And the people who got placebo was 18 months. Now remember, the treated group with Octreotide was 14 months, and here the group that got nothing is 18 months. So go figure. That's why I said it's very difficult to interpret the prior study. But in any case, here we are. Now we have long-term follow-up data from this that shows that the actual period of time of cancer control was 38.5 months before cancer started growing on the average, but for intestinal carcinoids, for neuroendocrine tumors of the small intestine, the average time until cancer started growing was 61.5 months. So this is the longest period of cancer control that people have reported with anything. That's even longer than people have claimed with PRRT. And this is with injections of Lanreotide once a month. So it's really remarkable how much mileage you can get out of this. And remember, these numbers are statistical averages. That means in a particular patient, half the people are doing better than that. And that's why some people are still taking this medication many, 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 many years later. Okay, Now, the other thing that we can do, and this is the result of some newer clinical trials that are particularly exciting, is using the somatostatin analog binding to the somatostatin receptor as a means of delivering a radioactive payload. Now, what you see over here listed as target, okay, is the somatostatin receptor on a cancer cell. What you see over here that says ligand is the thing that sticks to the target. That's the somatostatin analog. That's medicine that's like Octreotide and Lanreotide. It's attached to a chelator over here where you put the radioactive stuff in it. If you put gallium-68 into this little cage, then you have gallium-68 for doing dotatate PET scanning, and we call that a reporting unit, the gallium-68, because it reports where the cancer is and shoots out rays that says, here I am, I'm a cancer. If you put in lutetium-177, which is made as a killer isotope, it'll go to deliver a payload to try to get rid of the cancer by killing it. That's lutetium-177. This whole thing is otherwise identical. And that's why if you have a positive gallium-68 Octreotide, gallium-68 dotatate scan, which is positive, you're extraordinarily likely to respond to lutetium-177 dotatate therapy, because the dotatate is exactly the same, and that's what's sticking to the cancer. Okay? So here goes. That's what happens when you inject it intravenously. It goes through the body. The moment it finds a cancer, it sticks. So this was done in Europe for probably a decade before finally a good randomized phase 3 trial was set up to try to prove with certainty that the stuff is working. And that was done. It's known as NETTER-1. Again, the title coming from initials of certain words to try to make it work, like the last one I showed you. This was a trial where half of the people got lutetium-177 dotatate. We call it 200 millicuries in America, but in Europe they call the same dose 7.4 gigabecquerels because they like to use different measuring systems. But in any case, it's the same amount of radiation as was being used in Europe, one time every 2 months times 4. So the whole treatment is 8 times, and it's a total of 4 injections. In the United States, we do it routinely as an outpatient. People tolerate it well if it's done in a by experienced people, and the side effects are anticipated or treated aggressively so that you don't get sick from it. And the main toxicity being nausea and vomiting from the original formulation, where very, very high concentrations of amino acids were being used to prevent kidney damage. And now, like they do at the NIH, we have a kind of amino acid formulation that does not cause nausea and vomiting. So it's really user-friendly now. In any case, this was the treated group, Octreotide, Octreotide with lutetium-177 for 4 cycles, and there was a control group that got a double dose of regular Octreotide. These people all had progressed on 30 milligrams of Octreotide when they went on the trial. So now the people that did not get the radioactive form got 60 milligrams of Octreotide LAR. Okay, so the question is, do we need to have the toxicities of radiation if a double dose of non-radioactive Octreotide is just as good or better? We don't know. So we did that trial, and the results were amazing. The people who got the lutetium-177 dotatate, which is the upper curve, the scale on the left is number of patients, and the one on the bottom is time. The average time to cancer starting to progress, which is what the trial was designed to do to keep the cancer from growing, was never reached at the time of this publication in New England Journal of Medicine. And in the people who got the double dose Sandostatin, the average time to progression was about 8 months. And you could see those patients progressed quite rapidly. It didn't do much at all to just double the dose. So if somebody's on 30 milligrams of Octreotide LAR, their cancer is growing. You're kind of wasting your time thinking just by giving 40 milligrams or 50 milligrams or 60 milligrams that you're going to make a big impact on the disease. You need to do something significantly more than that. The latest follow-up from that study is still not reached the average person relapsing yet, because most people have never had a relapse. But if you extrapolate that upper curve, it looks like it's probably going to control the cancer for around 40 months or so, which is quite respectable considering they've been resistant to so many other treatments before they ever got PRRT. PRRT is very good for properly selected patients. This is the item that we call response rate. And this is something extremely important because people have the feeling that if they get PRRT, the cancer is going to get nuked and it's going to be dead, and you want to get PRRT right away, let's go sign up for PRRT because that's going to solve the problem. But remember, the goal of the PRRT trial was progression-free survival prolongation. It was trying to keep the cancer from growing. If you look how many patients actually had a tumor shrinkage, it's actually not that many. There were 19% of the patients had a tumor shrinkage of 30% or more, which was considered a significant reduction in the size of tumor in 19%. And the other 80% roughly did not have a significant shrink of tumor. It was either the same or just minimal reduction. Most people it was able to control, but not everybody. So that's why this treatment needs to be put in its proper perspective and done at the right time for the right reason, because like any treatment in cancer, it does have its own side effects. The toxicities are usually tolerable and not a big problem, which is why we give it routinely as an outpatient. Now that we have amino acids that don't make people sick with nausea and vomiting, it makes it even easier to tolerate. Not everybody is doing that, but I hope it's going to become very widespread to use the new amino acid formulation. And it doesn't cause too much nausea, but it can cause some. It can cause fatigue. It can make blood counts get low, usually not severely low, but it can. In the older age group, blood counts get lower than otherwise, particularly the platelet count. Some people need transfusions, and some people can have deterioration of kidney function, which is usually temporary. Some people can have deterioration of liver function, particularly if they've had radioembolization before. So I think that it needs to be done with care. And you always have to say, what are the alternatives? What are we treating? What is the best thing to do at this time? As I mentioned earlier, and that's why we have a multidisciplinary team. Should we embolize the liver? Should we do surgical removal of tumors? Should we do PRRT? How widespread is it? What other treatments has the patient had? What are the goals of treatment? You have to put it all together to individualize, but it's a wonderful extra treatment. In addition, quality of life has been studied extensively with PRRT. This was a paper we published last year in Journal of Clinical Oncology. It turns out that after getting 4 cycles of PRRT, most people have improvement in quality of life. And as you can see, it reduces diarrhea, reduces flushing, reduces pain, improves energy level, improves what we call performance status, which is people's functional level, and it improves a global health score based on patients' evaluation of their state of health. All these things are improved. However, in the first 1 or 2 months after PRRT, the physical condition can dramatically worsen because you get swelling and edema from the inflammation associated with radiation. When you get the tumors nuked, they can get very, very inflamed. And as a result, there can be some swelling temporarily, and that can worsen pain and worsen symptoms of carcinoid syndrome and worsen diarrhea. So these things can worsen in the first month or 2, and then after that, they just tend to get better and better and better and better. So you have to be prepared to deal with this with extra somatostatin analogs, extra ancillary medications for treating symptoms. Be prepared so that when these things happen, it's expected and you're ready for it. And then after that, things start getting better and better. And then you say, wow, I'm not taking pain medicine anymore. I'm really happy. But in the beginning, it can be hard in the first month. Okay, who should be treated is an area of some controversy, but I'm going to give you my take on the situation. One, presence of metastatic neuroendocrine tumor in the gastrointestinal tract or pancreas. This is what has been approved by the FDA. In some ways, we should be happy that we have this approval from the FDA because the clinical trial that I showed you that led to the approval, called NETTER-1, just treated people with intestinal carcinoids. But along with the presentation to the FDA on intestinal carcinoids as backup data, data on roughly 2,000 European patients with all kinds of other neuroendocrine tumors was submitted, showing effectiveness even though it wasn't randomized. And on that basis, the FDA approved neuroendocrine tumor treatment for with PRRT for anything that started in the gastrointestinal tract or pancreas. However, they made that distinction and used the word gastroenteropancreatic neuroendocrine tumor. As a result, all of the folks who have lung carcinoids, for example, and other sites of primary are often being denied by insurance companies. And that's really a problem because the treatment itself is really what we call site agnostic. In other words, if you have a positive gallium scan and you have strong somatostatin receptors, it makes absolutely no difference if it started in your head or your toe. As long as the medicine is able to get in there and you have high binding, it's going to probably work just fine. And there's lots of data from Europe about that, and there's a small amount of data about that from the United States. But since we don't have that approval, it makes a big challenge for patients who have primary tumors in those other sites. And sometimes we get approval anyway, and other times it may be most appropriate to treat these patients on a clinical trial, which will allow lung patients to go on. And the trial Dr. Rivera was telling you about an example of such a trial where patients can still be given an opportunity to have PRRT when otherwise they might not have such an opportunity. Well-differentiated histology typically is associated with tumors which are not as fast growing. All the clinical trials that were done leading to approval were with well-differentiated neuroendocrine tumors. Basically grade 1 and grade 2. However, the approval does not mention grade. So as a result, even if you're grade 3, you could potentially be treated with PRRT as a standard of care treatment. But in actuality, the people that do the best with grade 3 are ones that have the grade 3 neuroendocrine tumor and not the ones who have the grade 3 neuroendocrine carcinoma, because they tend to grow so fast that you don't have the ability to treat with a slow-acting treatment like PRRT. But that has to be individualized, and there is some data on using PRRT in the more aggressive treatments. In the United States, people tend to have previously been treated with a somatostatin analog and progressed on that. They have progressive disease on imaging, on MRI or CT. They have a positive gallium-68 dotatate scan. They have to have adequate renal function because the lutetium-177 dotatate that is not bound to the tumor has to get urinated out. It gets eliminated in the urine. So if you don't have good kidney function, it can hang around in the body, lead to high blood levels with lots of toxicity, and can end up destroying the kidneys. So that's really important. So before anybody is treated, they need to have a blood test of their kidney function, and the creatinine level should not be more than 1.7, or an estimated creatinine clearance of should not be below 50. They should have good enough performance status and physiologic function, in other words, things like liver function and lung function and heart function and whatever, that they can tolerate a relatively aggressive treatment. And at least in our center, everybody who gets treated with PRRT is discussed at our multi-center, multidisciplinary tumor board, and we all agree that this is the best next step and the patient would be appropriate. Now, clinical trials, which everybody here is very interested in, there are lots of new trials for PRRT, and if you don't get PRRT today, maybe you'll get PRRT in 6 months, or you get PRRT next year, or you get PRRT in 2 years, and whenever you're getting it, there's going to be so much more we're going to know about PRRT, and so many more PRRT trials are going to be available because it's a rapidly changing field. So just some of the interesting things that are being done. People are using new ligands, in other words, things that instead of a somatostatin analog, we're using other molecules to stick to the somatostatin receptor, like antibodies to the somatostatin receptor, antagonists of the somatostatin receptor instead of so-called agonists. One that's in trial right now is called JR11. It's quite promising. We have to see how all of these things develop. People are using PRRT into the hepatic artery to do intra-arterial PRRT, an interesting concept since it doesn't involve blocking off blood vessels in the liver like chemoembolization and bland embolization and radioembolization, but you can have a very high dose of drug delivered into the liver. PRRT is being used in combination with other drugs that not only are active in neuroendocrine tumors, but can be synergistic and make them work better. They're so-called radiosensitizers. PRRT can be used potentially with immune therapy, and there are clinical trials like that, trying to make the immune therapy more effective after you cause mutations in the cancer from the radiation of PRRT. People are doing studies with new isotopes. Instead of using lutetium-177 that shoots out a beta particle, people are doing studies with alpha particles, which have potentially less collateral damage. Lutetium-177, by the way, has very little collateral damage. It shoots out a ray that only goes for 2 millimeters. A beta particle travels 2 millimeters from lutetium-177 as opposed to 12 millimeters from yttrium-90. It's much more damaging, but alpha particles much less than that. So we have to see how that turns out. And new targets beyond the somatostatin receptor. Not everybody has somatostatin receptors. And it could be that there are other molecular targets, and we're already thinking about things like that and working on trials for that. So this is a very exciting area. I'm very, very excited about where we're going. And just stay tuned, as these things develop. This is an example of the remarkable effectiveness of using gallium-68 dotatate PET as a means of predicting response and following response, and lutetium-177 dotatate as a therapy. This is from Richard Baum in Bad Berka, Germany, a CAT scan showing 2 tumors that are positive on the gallium-68 PET. You can see them sort of on the CAT scan, and they light up like little light bulbs when you superimpose the gallium scan and the CAT scan on top of each other. You treat with 1 cycle of PRRT. The CAT scan looks pretty good, but the tumors are still there, lighting up nicely on the gallium scan. 1 more treatment. They're gone on both. The patient's clinically now in a remission. This patient did not go on and have 4 treatments, which is what we're currently recommending, but actually had only 2. And a year later, it came back, but the first place you see that recurrence is in the liver. It's not on the CAT scan. So CAT scans are very, very good for measuring the size of tumors, but for sensitivity and picking things up, and for determining responsiveness to treatment with radioactive material that relies on somatostatin receptor binding. There's nothing like a gallium-68 dotatate PET. Okay, we did a study in California right before I came out here on 100 patients who had already had all the standard imagings with MRI and CAT and somatostatin receptor imaging without including gallium-68. They had Octreotide scans, and then they had a gallium-68. It changed the management in 60% of patients. You'd say what you wanted to do before the patient had the scan, and say after the scan, how did it change management? 60% of the time. So that's really a major, major advance in the field, and again, why we like it. The next molecular target I wanted to mention is the so-called mTOR, an enzyme inside of neuroendocrine tumors. And it turns out there's a remarkable story that we don't really have time to tell today about how this was developed, but maybe I'll just give you just a little insight into this. There was somebody who did what under normal circumstances would be a completely crackpot science. He went to the Easter Islands to, I guess, look at the giant heads made out of stone. They have stone warriors standing there in the big island in Easter Islands. It's called Rapa Nui Island. Okay, and while he was there, he got this idea that he ought to take some dirt back. I don't know if he had a Ziploc bag or something, but he put some of the Easter Island dirt in a bag and took it home to analyze. Because he got this idea that there might be a microorganism in that dirt that had never been discovered before, and maybe it's making an antibiotic that was never discovered before. And I guess he'd be rich and famous and have a new antibiotic. It's like finding streptomycin coming from Streptomyces. So he brought this dirt back from Easter Island because it's such an isolated ecosystem that maybe it would be something different that wouldn't be in China and Brazil and Africa, wherever, where people have looked all over the world for materials making antibiotics. So what happened? He hit the jackpot. There was a new antibiotic. He named it after the island. It's called Rapa Nui Island. So he called his antibiotic rapamycin. So rapamycin comes from Rapa Nui Island. So then they looked at the cell to see what this antibiotic was doing, and they found out that it was working on an enzyme that had never been discovered in the world. So they had to sound a little scientific when they didn't know what they were doing. So they called the enzyme mTOR. That stands for mammalian target of rapamycin. So now you know exactly what it does. It's an mTOR inhibitor, and it inhibits mTOR. And what does mTOR do? It sits there and gets inhibited by rapamycin. Right at that time was the NIH having the equivalent of today's moonshot. At that time, they thought they would try to find cures for cancer by a brute force approach, where every time anybody would ever discover a new organic chemical, they would put it through a high throughput screening system of cells and culture and see if the new chemical had the ability to kill cancer cells. So this chemical got fed into this along with maybe 10,000 other chemicals. And it turned out it was a fantastic anti-cancer drug. Okay, so then people got really excited and started doing lots and lots of research about how this would work as an anti-cancer drug. And it turned out that mTOR is a central regulator in the cell that determines how long the cells live and how much they eat and how much oxygen they take up, and it runs the show. And it turned out that neuroendocrine cancers have mutations in the mTOR pathway system that tremendously increases the amount of mTOR. So when you use an inhibitor, you selectively knock off neuroendocrine cells. So based on that, some trials were done with these mTOR inhibitors. Rapamycin, by the way, has a generic name of sirolimus. That's the name of rapamycin. So a pharmaceutical company made everolimus out of it. It's a user-friendly form of sirolimus. It's sort of like prednisone is a user-friendly form of cortisol. Okay, so that's how we get everolimus. It's a natural substance, basically. And it works as an antibiotic on some microorganisms. So a randomized trial of everolimus versus placebo in pancreatic neuroendocrine tumors goes by the nickname of RADIANT-3, the largest trial in the history of the world in pancreatic neuroendocrine tumors. This particular neuroendocrine tumor has an incidence of 3 in a million. It's much less common than intestinal neuroendocrine tumors, even though, I don't know what all the noise is. In any case, for those of us doing neuroendocrine oncology see this as one of our most common diseases every day, but in the real world it's not very common. And what happened is that, thank you. Is this better? I'll turn off the light. Is this better? Okay, so we won't have so much effect. Okay, so here we are with back to RADIANT-3 clinical trial of everolimus versus placebo. And again, done at a time when this was felt to be an ethical way to do a trial. And there was a crossover, so anybody who did not respond was able to be treated with the everolimus. The results were extraordinary. The progression-free survival dramatically improved, the upper line in red. And within months, it was FDA approved, and now we have everolimus for pancreatic neuroendocrine tumors. A similar trial was done in neuroendocrine tumors in the gastrointestinal tract and lung. That's called RADIANT-4. And again, results are practically the same. It works very, very well. The period of cancer control on the average is not as long as it is with PRRT, but some individuals, it can go for years, and it's if the dose is adjusted right, it's very user-friendly. The key is adjusting the dose right. The doctor gives you a prescription for the approved 10 milligram dose and tells you to come back next month, and you come back a month later in a wheelchair with overwhelming fatigue and diarrhea and skin rash and mouth sores and say, I'm never going to take this rat poison again. It wouldn't be very good. But if you understand that if you take it and you tolerate it fine, that's great. But if you don't, you stop it immediately, wait for the side effects to resolve, and then you start at a half dose. And then if side effects happen again, you immediately stop, wait for side effects to get better, and then go to half a dose again. So the approved standard starting dose is 10 milligrams a day. But there are plenty of people who need 5 milligrams a day or even 5 milligrams every other day. It just has to be used properly, like all medications, really. But with everolimus, it really is dramatically different in toxicity depending on the dose. There's a new generation of mTOR inhibitors. This is one I've been particularly interested in, CC223, and there are other ones, MLN0128. These are what we call dual kinase inhibitors that are
next generation after everolimus. We have to see where that goes. Hopefully it'll be a major improvement. We have another kind of a pill called Sunitinib, Sutent. This was one of the randomized trials that also I had on the list that led to rapid FDA approval. It's a completely different drug than the mTOR inhibitor. It works on the vasculature of cells, but on the blood supply. But the results are almost superimposable in terms of progression-free survival, and the side effect profile is also generally tolerable if the dose is adjusted right. Now, one clinical trial that's open right now that I'm very excited about, and we are using at Mount Sinai a lot, is with cabozantinib. Cabozantinib is an inhibitor of blood vessel growth like Sutent, like sunitinib, but in addition inhibits something called c-Met, which is a very important enzyme in controlling neuroendocrine tumors. And this drug has turned out to be extremely useful. This was a phase 2 trial that was done in neuroendocrine tumors, and the people who had treatment had very, very long periods of cancer control with both pancreatic neuroendocrine tumors, which are green, and carcinoid tumors, which are white, close to 2 years of control or more with a pill with generally very tolerable toxicity. So this is an extremely important drug. Based on that, a randomized clinical trial has been set up, and that's what we're doing right now, which was a study that was done, that is being done, where people are being treated with cabozantinib. 2 people get cabozantinib, 1 person does not, 1 person gets placebo, 2 people get cabozantinib. This is after you've already had normal treatments like somatostatin analogs and everolimus, and you need another treatment. This is a terrific treatment. It's considered by the National Cancer Institute to be one of the highest priority trials, which is why it's being sponsored by the cooperative group mechanism, and it's available with free drug for anybody who qualifies. And I think it's an excellent trial. If somebody progresses on cabozantinib or is the 1 person out of 3 who doesn't get it and has progression, they go off, of course, and can still fall back on other things. But it gives an opportunity to be treated with a very tolerable and effective treatment, which, by the way, has been approved for a neuroendocrine tumor of the thyroid gland called medullary carcinoma of the thyroid, and it's also approved for kidney cancer. But it's really going to rely on this trial to get it approved for neuroendocrine cancer. So being in this trial not only benefits the person getting it, but also will benefit the whole world of neuroendocrine patients. I think I'll whiz through these because they're not quite as relevant right at the moment, although very interesting trials. I wanted to bring your attention to a trial that was just completed in the past year of temozolomide or Temidor plus capecitabine, also known as Xeloda. And it goes by the nickname of Cape-Tem. You might have heard of that one. It's a combination of 2 oral, generally pretty well tolerated chemotherapy drugs for pancreatic neuroendocrine tumors. Okay? It was a randomized trial of temozolomide alone, which some people felt was just as good as the combination, versus the combination of temozolomide Xeloda, known as Cape-Tem. Okay? This was just presented last year. 1/3 of the patients had a major reduction in the size of their tumors, which was a remarkable, slides are messing up a little bit, a remarkable reduction in the size of their tumors. About 1/3 of the patients had a partial response by so-called RECIST criteria, and most of the patients, the cancer just stopped growing for prolonged periods of time. So based on that, a randomized trial was done, and it turned out that the combination is better. So if somebody has the opportunity to be on temozolomide alone versus temozolomide in combination with capecitabine, the combination is the best. Cape-Tem, known as Temidor and Xeloda in the brands. Okay? Now, carcinoid syndrome, you've been hearing a lot about today, and a few minutes, I could talk about carcinoid syndrome. Subcutaneous Octreotide, this is the original trial that proved that Sandostatin works in carcinoid syndrome. You can see that flushing and diarrhea have been dramatically reduced when people were treated with Octreotide immediate release. When you use Octreotide LAR, this is the study that proved that the combination is really good. That 5-HIAA fell by roughly 50%. Diarrhea went way down. Flushing went way down. That little peak you see right before was sort of a washout from the subcutaneous Octreotide. They stopped everything. Everything got terrible, and then you treat it with the drug, and everything got better. So it's a very good treatment. It got approved, but that approval was for flushing and diarrhea. Also, a trial was done with Lanreotide for treating carcinoid syndrome, diarrhea, and it turns out it works perfectly well. In addition to treating cancer, it also treats diarrhea. So for the treatment of carcinoid syndrome, we have Octreotide immediate release, Octreotide acetate, we have Octreotide LAR, and we have Lanreotide. The Lanreotide trial had to be a little bit more complicated because these people were all on subcutaneous Octreotide, and then they got either Lanreotide or placebo on top of it, and the goal of the trial was to reduce the need for subcutaneous Octreotide injections. Okay, so if the Lanreotide works, then you don't have to use rescue shots anymore, and that's indeed what happened. Okay, using high-dose Octreotide as an anti-cancer drug is not very good. As you can see from that NETTER-1 trial, control group giving 60 milligrams of Octreotide didn't do much for controlling cancer. However, if somebody is having persistent flushing and diarrhea and needs better control, sometimes going to higher doses of Octreotide is better. This is what is meant by above-label Octreotide. That means above the dose which is on the package label and is FDA approved. So we can't really recommend it, but I could say that it has potential for efficacy. However, before going to that, normally we would use Talotrostat, we would use subcutaneous rescue injections of Octreotide, and we do other maneuvers. This is Talotrostat inhibiting the tryptophan hydroxylase, which you heard about, and cuts down serotonin production. In the Telestar study, Dr. Zatz had already talked about. I don't think I need to go over this again. Okay. Interestingly, there's an anti-nausea medicine known as Ondansetron, brand name Zofran. This is a very common medicine for nausea. And it turns out the way it works on nausea is by antagonizing gut serotonin. And if you use very high doses of this medication, even though it's not approved for this purpose, it looks like it can control carcinoid diarrhea where other things aren't working. So in addition to Octreotide in various forms and somatostatin analogs that we talked about, and Talotrostat, and using steroid hormones like Solumedrol, Ondansetron is another thing, and I've treated intraoperative carcinoid crisis situations with very large doses of intravenous Ondansetron in addition to the things that Dr. Zatz was talking about. Just a reminder that the NETTER-1 trial demonstrated an improvement in quality of life. I showed you earlier in reduction in diarrhea and flushing, but in particular carcinoid syndrome diarrhea gets much better with PRRT. This is the picture of what happens with the diarrhea. And when people got the PRRT, these are the two arms in the NETTER-1 trial, much better than the people that got 60 milligrams of Octreotide were the people that got the radioactive kind. Carcinoid crisis, I think, has been very nicely discussed. Now, how do we figure out what to do? This is really a challenge because now we have gone from no treatments to a whole bunch of treatments. And this is what we have on our plate. For midgut primary tumors, meaning generally intestinal carcinoids, okay, we have Octreotide and Lanreotide, the various somatostatin analogs in the different formulations. We have everolimus. We have interferon, which has been shown. I glossed over one of the slides more quickly, but it does have activity in neuroendocrine tumors. Used more in Europe than in the United States, but it is available. PRRT, Talotrostat for carcinoid syndrome. Just observation in very highly selected patients. There are some people who have tumors that are like a 16th of an inch across and haven't grown in the last 5 years. And it's hard to say, boy, you have to start somatostatin analog therapy right away. Maybe they'll go for another 5 years before you even need it. Some people have really, really, really slow-growing tumors with very, very low tumor burdens. So those people might be watched without any therapy at all. That's an option. And always clinical trials are an option. Whenever you think about therapy, you say what is approved and what's on trial and what trials are reasonable. And sometimes the trial's the way to go. Huh? Oh, okay. It was a fly. Okay, I thought you were signaling me. Okay, pancreatic neuroendocrine tumors, we have all of the same things, and the addition of sunitinib, which has been approved, Sutent, and the addition of chemotherapy with temozolomide and capecitabine, not approved, but clearly demonstrated to be highly effective, and both drugs are on the market, and the chemotherapy, which is approved but is never used hardly, and that's streptozocin. It's a very, very old chemotherapy with a lot of side effects, including potential for kidney failure, and it's not used so much anymore. And in addition to all of this, remember that carcinoid syndrome can also be treated, and carcinoid tumors, with surgery, with embolization, with ablation, with all kinds of other treatments at the same time. And every time we make a decision about a systemic treatment, treating the whole body, you have to say also what should be done or could be done. That involves liver if that's the major site of disease. Okay? Now, this is just a summary of a lot of things I talked about, just to give you some idea how we think about these things. The extent of the tumor, is it localized or is it metastatic? If it's localized, surgery is overwhelmingly the best thing to do. If it's metastatic, more things are on the plate. Site of metastasis, if it's liver only or liver dominant, then we have a whole big range of liver-directed therapies. If it's involving the whole body in a major way, we don't have those options. We have to just go straight to things that treat the whole body, like various types of biologics and chemotherapy and PRRT. What does the pathology look like? Is it a well-differentiated tumor or a poorly differentiated tumor? How fast is it growing? How fast is it growing is very important, because you always have to use your head, no matter what the pathologist tells you. If you have a biopsy of a pancreatic tumor that's done with a fine needle aspirate at endoscopic ultrasound, and it says well-differentiated low-grade neuroendocrine tumor, and then you look at an X-ray, a CAT scan 2-3 months later, and the tumor's doubled in size, that's not a slow-growing tumor. That's a fast-growing tumor, and it doesn't matter what the pathology says. The reason it came back low-grade, well-differentiated tumor was they got 2 cells in the needle when they did a fine needle aspiration, and the rest of the tumor is pretty nasty. Or it had some nasty areas and some not nasty areas. So you have to say, wait a minute, do I believe that pathology? Do I need another biopsy? Do I have to do something different? But you're not going to continue using a therapy that's not working if it's growing fast and resistant and not behaving like you would expect. Okay, primary site, pancreas is uniquely sensitive to all kinds of chemotherapy, immune therapy, not immune therapy, but chemotherapy and biologic therapy. And if cancers are making a hormone and causing a syndrome that needs to be treated more aggressively, as you heard from earlier speakers, people that have carcinoid syndrome that isn't controlled have a much worse prognosis and have to be treated more aggressively to get those symptoms better. And the same thing if cancers are making other hormones, pancreatic hormones or lung cancer, carcinoids are making hormones. You have to treat these things aggressively. And it doesn't matter which hormone. There are lung carcinoids that are making ACTH, cause severe Cushing syndrome. That needs to be treated very aggressively, or people end up dying of the complications of the hormone before you get a chance to treat the tumor. So you have to put everything in perspective and put it all together to optimize the treatment. Always remember the things on the bottom here, balancing the anticipated outcome with toxicity, thinking about the chance of achieving stable disease versus response, and what difference does it make. For example, if you have a pancreatic neuroendocrine tumor which is just on the verge of being operable but the surgeon isn't sure he can get it out, you can maybe treat with temozolomide and Xeloda, try to shrink it up, and then after it's shrunk up, go to surgery, because that treatment gives you the biggest chance of a major shrink, right? Otherwise, you might just start therapy with a somatostatin analog, or use everolimus if you don't have somatostatin receptors, or whatever it might be. You see, you have to say, what is the goal? In that case, the goal is tumor shrinkage. If the goal is long-term control, it's a different sequence of drugs. And you always have to say, what are the potential benefits and what are the potential risks? And if you have 5 treatments that all have a good chance of working, but one of them is way better tolerated, use the one that's way better tolerated now and don't become all debilitated and full of side effects. And by the time the well-tolerated one stops working, hopefully we'll have other things on the market or other things available investigationally, and maybe you'll be able to do a whole lot better and save things that are more toxic for another day. Okay, so that's the end of my formal remarks. Thank you very, very much.